In this video, we'll discuss Fick's law and the implications that Fick's law has for organisms. Fick's law is shown at the top here. It is a calculation expressing the relationship between the components of gas exchange. It doesn't only apply to biological situations, that's why I phrase it this way. And there are different ways to express Fick's law. The version we're seeing here is relatively simplified. And Fick's law has a lot of important implications for how organisms get their oxygen. So what Fick's law ends up meaning for biology is that there is a limit on the rate of exchange, which is Q, that's what we're interested in. It matters what substance you're getting your oxygen from, and so that would be either air or water. The surface area of the tissue where the exchange of oxygen from the air, from the environment into your body is important. And the concentration gradient of oxygen between the air or the water and your blood or hemolymph or tissues or whatever is important. And so again, this is the difference in the concentration, or really the partial pressure of oxygen in the air or water, depending on what the organism is breathing, and the tissues that are receiving the oxygen. And so that gradient, you want that difference to be as great as possible to maximize the rate of exchange. And finally, Fick's law means that the thickness of your gas exchange tissue should be pretty thin in order to get the highest rate of exchange. So there is a video here which I recommend watching, which talks a little bit more about how Fick's law can be used in terms of the going over the math, and also just in general the implications of Fick's law for non-biological situations. So I do recommend you watch that. And I'm going to talk a little bit more about how this applies to biology and gas exchange organs in particular. So in general, it's better to breathe air than water if you're trying to get oxygen. And that is because there is a higher partial pressure of oxygen in air than there is in water, especially if there is warm water that you're living in. And so this graph here shows that as the temperature increases, oxygen tends to go out of the water, so it will actually dissolve or evaporate, I guess dissolve is the right word, into the atmosphere or the air. So when I say air during this lecture, I mean the atmosphere, not oxygen in particular. Because oxygen in particular here we see decreases with increases in temperature, and so that's going to make it more difficult to breathe and more difficult to get oxygen from the water if there is less oxygen in the water. And organisms don't experience the same relationship if they're breathing air, because the oxygen stays in air even at high temperatures. Another implication of Fick's law for organisms in both air and water is that getting rid of CO2 is pretty easy. So we need to get oxygen in, and then the byproduct of our cellular respiration is carbon dioxide. And it's easy to get rid of carbon dioxide. So that's not too much of an environmental pressure or selective pressure in how organisms evolve. And we can see here for the case in terms of the atmosphere, the relative partial pressure or the composition of the atmosphere in terms of carbon dioxide is pretty low. And so it will always be easy for organisms to dispel carbon dioxide as they're exhaling or as they're releasing that molecular waste. We talked about this a little bit already, but in order to get the most oxygen out of the air or the water, you want to maximize the gradient. And so part of this is the carrying the oxygen away from the tissue or away from the site of gas exchange. And so we do this with blood, things like other organisms such as the lobster, I think that was in one of our images, they carry the oxygen away in something called hemolymph, but same kind of idea. We have a moving substance, a flow that is taking the oxygen away from the site, keeping the concentration of oxygen pretty low in this tissue so that oxygen is more likely to continue to diffuse into this tissue. And sorry, I should have given us some perspective here. So this is a blood vessel. This is an alveolus or a single chamber which air flows into. This is very small in scale, and we have many of these within our lungs. And we see here that carbon dioxide is also diffusing out here. And as we discussed, it's pretty easy to keep carbon dioxide flowing out into the atmosphere or the water, as the case may be. Now we have this, these last 2 points, which are that exchange tissues should be as thin as possible, which also tends to correlate with increasing surface area. And so this is what we see in many gas exchange tissues. So on the previous slide, we talked about this alveolus and how there are many small chambers like this in our lungs. So that would be increasing surface area if we have a lot of these, and increasing the surface area relative to the volume. And here we see these structures that are in the gills of a fish, for example. They have a lot of surface area in these folded lamellar structures. And we have folding here, and then we have the protrusions here so that these are all next to each other, creating a really high surface area. But they're also very thin to allow the greatest amount of contact with the environmental source of oxygen, which is water or air, coming in contact with the tissue as possible. So again, increasing contact between tissue and the water or air, and this allows for these tissues, or I guess selects for tissues through evolution that are both thin and have a high surface area. I also wanted to point out this image on the right hand here. So this, instead of showing the surface of these lamellar folds, it actually shows the inside of them. And we see all these little holes. So not only are these very thin, not only do they have a very high surface area, but in addition, there are multiple of these holes, these places, again, where gas exchange is occurring. And I encourage anyone who's interested to look at this website, which has more of these types of electron micrographs. Technically, they're not scanning electron micrographs because they're looking at the inside of the object rather than the outside. And so I think that wraps it up for this video, and the next video will be comparing the different types, or some different types, of gas exchange organs.